exogenous chemical that we can add to cells and sort of modify the behavior of our genetic circuit. Having these external control points allows us to bias our output signal and thus modulating our system to compensate for any uncertainties that we may have in our mathematical modeling and our mathematical predictions. In addition to that, all the different control points that we have in our circuit are also used as variables in our modular testing. The third and final feature in our design is construction flexibility. And what I mean by that is our team has deliberately chosen components that have functionally analogous parts in the registry. What this means is that if, say, one or two of our components aren't working so well in our system, we don't have to go through a radical redesign of the entire project. It's easy for us to simply go back to the registry, find something that is functionally analogous, and use that part to continue our project. So not only did we plan for that this year, the fact that we had this backup plan actually benefited our project. In our original project, we had an intermediary molecule in our system, which was the CI molecule that wasn't working very well. And because it wasn't working very well, all we had to do, because we had our backup, all we had to do was go back to the registry, find something that is functionally analogous, and we ended up finding the tetR system, which worked much better without making major changes to our design. So our three features in our design allowed us to gain some understanding in some details of our genetic circuit, and it also gave us some contingency in the case when one or more parts don't function the way that we expect them to. So keeping in mind of the design criteria that we have chosen for ourselves, we came up with this final design. And what we have here is a master switch, which is a pBAT promoter controlled by a Rabinos. We have a LacI inverter, a tetR inverter, and two reporters, which are the green and red fluorescent proteins. So conforming to our design criteria, if we look at the master switch as well as the two inverters, we see that the last piece on all three of these parts is a promoter. What that means is that it's really easy for us to just tack on a reporter to the end of those constructs, and we can basically test our circuit in different modules. This design has three different control points. We can add a Rabinos or IPTG to control the level of LacI, which is an intermediary molecule in our circuit, or we can add ATC to control the level of tetR, which would be another control point in our circuit. So now that you know what our circuit looks like, we're going to go ahead and explain to you how our circuit works.
So our reaction mechanisms is as follows. Andy mentioned that with a Rabinos addition to the gene construct, it would induce the promoter pBAD to create LacI. Now LacI actually is, its normal form is in dimer. So in the cold state, these dimers would be able to bind together to create tetramers. These tetramers would then bind onto repression sites, specifically the repression site right before the RFP, red fluorescent protein, which then causes the inhibition of red fluorescent protein to be expressed. And also it binds onto the repression site of tetR, which inhibits tetR expression. Now it is important to know that tetR is actually our inverter of this gene construct. What it exactly does is that if tetR is actually expressed, it binds on to the repressor site of GFP, which then causes GFP to not be expressed. In this case of cold temperatures, this tetR is not expressed, so therefore green fluorescent protein is expressed. In the alternative situation, when it is hot, what happens is these dimers never form tetramers. In fact, and because of that, it wouldn't be able to bind onto these repressor sites right before RFP. That means that red fluorescent protein is created. You can see red. And also, it would not bind onto the repressor site before tetR. tetR is then created, and it would bind onto the repressor site of GFP, causing inhibition of green fluorescent protein. So what that means is when it's cold, it's actually green. And when it's when it's hot, it's red. So I'm going to give it to Anne now to talk to you about the deterministic model.
Thank you, Wingyi. So here we have our model for our system. It's a shortened version of our complete model. It contains 5 state variables, as opposed to our complete model, which has 18, and that's posted on our wiki page. As you can see, it's a deterministic model. It's a deterministic model, and for our promoters, we used a Michaelis-Menten model. And so here, in our complete model, the rate order of our system is 18, and we've explored the system using various parameters, some of which we know, such as the half-life of RFP and GFP, and others we've estimated. So here we have the results from our model. As you can see, this is a graph that shows the relationship between the concentration of the reporter genes to the temperature. So on the x-axis, we have our temperature, and on the y-axis, we have our concentration of the reporter proteins. And the red line represents the concentration of RFP at certain temperatures, and the green line represents the concentration of GFP at certain temperatures. Now, if we look at the range between 27 degrees Celsius and 37 degrees Celsius, we see that there's a large change in the concentrations of the reporter proteins. Also, if you take a look, as the temperature increases, the concentration of RFP also increases, but the GFP decreases. So the range 27 degrees to 37 degrees is pretty important in this experiment. So also, it's also, I guess, important to note that cell growth and cell division were not accounted for in this model. This may mean that in reality, we would have a more gradual change in concentration as opposed to what is shown here. So that being said, we also decided that we did not want to use a stochastic model because most of our the concentration of our key players, such as temperature-sensitive LacI and tetR and GFP and RFP, were pretty large enough that using a deterministic model would have been sufficient. So now I'm going to hand it over to Andy, who's going to talk about how we constructed our device.
So our construction consists of a total of 5 ligation steps. These 5 ligation steps can be carried out in parallel. The way we designed our process flow is in such a way that some of the intermediary parts that we have are actual modules that we can use for testing. The entire construction process took about 3 months, and at the end of that, we actually do have a completed circuit that we designed at the beginning. This year, our team constructed a total of 15 new parts. 5 of those parts went directly into our completed genetic circuit. The other 10 parts, some were intermediary testing parts, some were parts that were at the middle of the project because they weren't functioning properly. So next, what we're going to show you is exactly what modules did we construct and their characterization. By Charles.
Thank you, Andy. Where's the pointer? Okay. Okay, so our testing phase pretty much consisted of 3 main modules. And each module is then which contributes to the overall characterization of our Celsius thermometer. So module number 1 is made up of a LAC inverter and a reporter. There are 2 versions of module 1, one with a GFP and one with an RFP on it. This is to just test both at the same time period, as opposed to one at a time in sequence. So what happens is pBAD is induced, which then produces LacI, which goes and represses our reporter. The purpose of module 1 is to test 3 things. Number 1 is to test the functionality of our reporters. Number 2 is to test the production of LacI. And number 3, and most important of all, is to test the actual temperature sensitivity of our LacI. So as you can see, okay, so this is the test for reporter functionality, and this is really focused on the latter half of this module. So on the x-axis is the IPTG, which is a small molecule inhibitor of LacI, and on the y-axis is normalized fluorescence. So as you can see, as with increasing IPTG, we see an increase in normalized fluorescence of GFP. The red solid line indicates a theoretical model output, whereas the blue dots indicate our experimental data points. There's analogous, we did the same experiment with mRFP and we saw the same curve and the same trend, so that data is not shown here. So this indicates that the production of IPTG, so this indicates that the functionality of our reporters is correct, they function correctly, and that the promoter in front of them also functions correctly as a result. Okay, second is to test the actual production of our LacI. So, as you said before, pBAD is induced by arabinose to produce LacI. With increasing LacI, we see a decrease in fluorescence of our mRFP. This shows that the production of LacI is concentration dependent on the arabinose. This shows that the production concentration of LacI is dependent on arabinose, and it produces a concentration dependent relationship. And finally, our temperature dependence. This was done at 27 degrees Celsius and 37 degrees Celsius, which was previously indicated in our theoretical model as the 2 extreme endpoints of our dynamic range. So as you can see, so at high temperatures, LacI repression is decreased, which is shown by this significant increase in mRFP at 37 degrees Celsius. So these 3 tests of module 1 show that each individual part functions correctly on its own as well as together as a whole. So at this point, we pretty much have a bare bone thermometer. So we like to take another step further by adding a 2-color measurement system. So this brings me to module number 2. Module number 2 consists of LacI inverter, a tetR inverter, and then a reporter. It's pretty much the inverted module 1. So pBAD induces LacI, which inhibits the tetR inverter, and the tetR itself inhibits the GFP. And this module 2 is just to test the tetR inverter. So again, at the x-axis we see ATC. ATC is another small molecule inhibitor of tetR. So we see that with increasing ATC, we see an increase in fluorescence. This shows that, again, the tetR inverter is working correctly. It also shows, indirectly, we can infer that the functionality of LacI inverter and the GFP have not been adversely affected by the introduction of this new tetR inverter between the two gene sites. And finally, module number 3 is pretty much our full Celsius thermometer. The correct behavior of this thermometer would be at higher temperatures, we get more and more RFP formation. At lower temperatures, we get more and more GFP formation. And this is shown by, again, 2 experiments done at 27 degrees and 37. With increasing temperature, we see that the ratio between mRFP and GFP increases. So based on all these graphs, these show that we have a basic functionality of our Celsius thermometer. And finally, this is a preliminary visual demonstration of our Celsius thermometer. What we did was to make a heat gradient, to create a heat gradient within a agar plate where the heat source is at the center, and then the heat will dissipate outwards to form a gradient, a radial gradient. And as you can see, there is some indication of two different colors being present, where in the middle it's all kind of orange and yellowish, and near the outer edges it's green. So this again provides additional support for the functionality of our Celsius thermometer.
So, after long hours and hard work in the lab, as can be seen by Charles's facial expression before an all-nighter and after an all-nighter, we can conclude that
So in conclusion, we have successfully completed the construction of our design genetic device. Along the way, we constructed 15 new parts and added them to the registry, many of which were characterized. In terms of functional components, we have completed a bare-bone one-color thermometer that is relatively well characterized on a part-by-part basis. In addition, we also have very strong evidence to indicate that our bare-bone thermometer has the correct functionality. Beyond our bare-bone thermometer, we have gone one step further and completed a full-feature two-color thermometer and gathered some measurement that show some pretty optimistic results to indicate that we have some basic functionality in our thermometer. And namely, what we see is we see both red and green fluorescence in our two-color thermometer. And we have some evidence to indicate that there is temperature dependence of these fluorescence levels. So before we end, we would just like to acknowledge.
we would like to thank the Engineering Society from University of Toronto and the Mathematical and Engineering Endowment Funds from University of Waterloo. But we give special thanks to all our faculty advisors as well as Professor Stephen Davies and Brian Ingalls for giving us so much support and advice on all of this. Without them, I don't think we can ever finish this in time. And of course, all the other people in our group and iGEM, of course, for allowing us to give this opportunity to do this. Thank you. Any questions? Yes. So the question is, for the initial design, have we considered HSOC, heat shock proteins, and cold shock, and pardon? And promoters. Right. Okay, so the question again was if we considered heat shock proteins and other heat sensing protein promoters. I think Andy would be best to answer that question. So.
I think the answer to that would be no, we have not considered specifically the heat shock promoter. What we have considered is other possible promoters for our system. For example, we have a pBAD promoter as a master switch that turns on our entire system. We have considered the possibility of using a different promoter that's constitutively expresses, has constitutive expression, which we leave our system on all the time. So I hope that answers your question. Okay. Yes. So the question is, in our model, it seems that we have a quite sharp change in our fluorescence levels with varying temperature. Have we thought of strategies to kind of level it out to be able to cover a wider range of temperatures? Would that be okay?
Okay, well, like, I mean, obviously, each of these experiments can be done again in a lot more depth and detail, and that will probably give us more insight as to what the true dynamic range is. We just tried two of them, just time limit. Oh, that was we measured. Okay, I had to kind of overcompensate for the heat resistance of the plate, but we put at 40. We put it at 37. Something we knew in the middle of the plate. We tried to get it to be 37. Well, we had no way to measure that. we had to kind of guess and try to get a nice even thing. But yeah, obviously we have to refine each experiment over and over again to actually really answer that question. Yes. What do we use as a heat source? Oh, the very last the plate. Okay, we used a we used a hot plate, and we put a coin on the hot plate and put the plate on top of it. So we get a conduction of heat in the middle, and we try to rely on sideways dissipation of the heat to cool down the outer edges of our plate. Yeah, it kind of worked.
Trial and error.
Yeah. Any other questions? Oh, yes, sorry. We already ran into that problem. Okay, so would there be a problem with cells growing at the lower temperature ranges? So obviously, I mean, they're biological entities, so they're a very limited range of temperatures which they can grow properly and function before the behavior between 37 degrees Celsius and 22 is radically different. Yeah, I probably wouldn't go below, just based on the two experiments we've done, go below the 27, because that even growth rates dramatically different with those difference in temperatures.
I'd just like to add to that answer. So, I mean, our temperature range is limited if we use E. coli, but in the future, potentially we can use other strains of bacteria that can survive at more extreme temperatures. And by doing that, we're able to expand our temperature range. That's just a thought.